Good morning, students. In last class, we studied about sexual reproduction in bacteria. In this class, we will study sexual reproduction in bacteria. First of all, sex organ does not have evolved in bacteria, but sexual reproduction occurs by genetic recombination, and there are three mechanisms. First is conjugation. Second, transformation. And the third one is transduction. So, we start with conjugation. Studied by Liederberg. Bacterial cells have minute hair-like projection known as villi on their surface. Some villi are greater in length and form conjugation tube during conjugation. A bacteria which have F plasmids apart from main genome is known as male cell or F cell or donor cell. The bacterial cell which lacks plasmids, F plasmids, are called female cell or recipient cell. So, the villi present over the male make a conjugation with F plus cell with F minus cell. At the same time, the F plasmids make its copy and pass into another cell. As a result, this male cell F plus and this one also becomes F plus. So in this process, the female cell converts into male cell or donor cell, recipients from recipient cell. So the change in genetic setup of a cell means genetic recombination, and genetic recombination has occurred, that means sexual reproduction has occurred. So, this process is called conjugation. It occurs in several unicellular eukaryotic forms in different ways, but it is very prominent in prokaryotic bacterial cells. So conjugation is clear. Next is one more thing. If F plasmid gets associated with the main genome, then such cells are called high frequency recombinant cells. When the F plasmid get associated with the main genome, such cells are called HFR cells. So F plus cells which possess F plasmid, which causes F plasmid, or fertility factor. Fertility. Conjugation tube. Conjugation tube is formed by sex plate. Conjugation tube is formed by sex plate. Okay, so this is all about conjugation. In this process, a connection tube is formed between two cells. One is male cell, another is female cell or recipient cell. As the connection is established, a copy of F plasmid passes into another cell, and in this way, genetic recombination occurs, and this mode of reproduction is called conjugation. So after conjugation, we will study about transformation. What is transformation? This principle was derived by Griffith about 1928. At that time, there was a big race among scientific community to find out the genetic material, the molecular identification of genetic material. What is that? Is that protein? Is that DNA or RNA or any other biomolecules? So, a disease was prevalent, which is known as pneumonia. Pneumonia is caused by Streptococcus pneumonia or Diplococcus pneumonia. Streptococcus pneumonia. Now, this had two variants, two types of strains. One was a rough strain and the other was a strain. The rough strain was non-virulent and this was virulent form. So, Griffith conducted an experiment. He injected S strain in mice. This is experiment. Step 1. S strain in mice. Mice died. Injected R strain in mice. Mice survived. That means R strain could not cause disease in mice. Then injected heat-killed S strain in mice. Mice survived. Then heat-killed S strain plus live R strain. Heat-killed S strain plus R strain. Now what will happen on the basis of this experiment? What should be happen? Heat killed SS strain in mice survived, that means no disease. And RS strain in mice survived, that means no disease. These two including here. So what should be the result? The result should be that mice should survive, but mice dies. Why? RS strain was non-violent and SS strain was violent. SS strain was heat killed and provided they could not cause disease in the body. Heat killed SS strain plus live RS strain was injected, then mice died because the disease clicked on. So who caused disease? The disease is caused by RS strain because he is only in the living condition, life condition. And heat killed SS strain is in dead condition. So what transformed the non-virulent strain to virulent strain? There are some chemicals, molecules, which is called genetic material. And the thing which passed from heat killed SS strain to RS strain is genetic material. So, conclusion. The chemical which passes from, something has passed from heat killed SS strain to RS strain, which has transformed it into virulent form. Some molecule has passed from SS strain, heat killed SS strain, heat killed SS strain to RS strain, and that molecule is responsible for transforming RS strain from non-virulent to virulence. So, transformation principle was proposed by Griffith in 1928 on the basis of experiment based over Streptococcus pneumoniae, which has two strains. One is virulent strain, the other is non-virulent strain. In his experiment, he found that when heat killed SS strain and live RS strain was injected into the mice died due to the disease caused by this combination. So how the RS strain become virulent? That means something has passed from heat killed SS strain to the RS strain, and the thing which has passed is the genetic material, because it is governing the nature of the RS strain. So the substance which has passed from heat killed SS strain to RS strain is genetic material. So Griffith could not provide the chemical evidence and could not analyze the molecule responsible for it. But after somehow 5 to 10 years after Griffith, some scientists like MacLeod, McCarthy designed the experiment for providing chemical evidence in support of Griffith's experiment. And what he did, combination, last one, we represent it by combination, plus proteases, then injected in mice. तो अगर वह पास होने वाला मॉलिक्यूल अगर प्रोटीन है तो प्रोटीन को अगर उसमें प्रोटीएसेस डाल दें तो दैट विल डिग्रेड प्रोटीन. So protein could cannot pass and disease cannot prevail or occur. Then he noted that disease caused. That means the passing substance is not protein. So mice dies. That means the protein is not responsible, protein is not passing. Then this combination, that means heat killed S strength plus live R strength is treated with RNAs. Then also mice dies. That means RNA is not passing. Then the same combination plus DNA. Mice survives. Mice survives how? That means disease could not be caused. Disease नहीं कॉज किया. क्यों नहीं कॉज किया? क्योंकि जो उसको वायरलेंट बनाता है वो डीएनए था और जब हम डीएनए इसमें शामिल किए तो उसने डीएनए को डिसइंटीग्रेट कर दिया, which could not pass into the live cell, and so that molecule which passed from virulent to non-virulent species is DNA, and that DNA is responsible for making the non-virulent strain virulent. So DNA is the genetic material. That could be established later the transformation experiment of Griffith in 1928. So we have discussed conjugation and transformation. The rest topic plus revision will be done in our next video or classroom that will be dedicated to transduction and revision of this chapter. Okay? Till then, stay at home, stay safe, do some creative work, note down the taught portion in your fair notebook, and rest is okay. Bye. See the board if you can. We have studied two things, conjugation and transformation. Next topic is transduction, in which we will study about lysogenic and lytic cycle of phages in bacteria. So, goodbye.